Hello children, today we will continue with the topic reproduction only, and a subtopic was left in that in the last lecture, and that subtopic was test tube baby or in vitro fertilization. Now, actually test tube baby is also called as in vitro fertilization, as test tube baby is the main achievement of biotechnology. It is the main achievement of biotechnology in human welfare. Now, the most important aspect of this technique is that the ladies deprived of children become capable of being pregnant. In this technique, what happens is the ovum is obtained. The ovum is obtained from female's body, and is fertilized by the sperm of the male under controlled conditions. Now, the whole process is completed outside the body of the female. One important thing about this is the process is completed outside the female's body in the laboratory. If the egg cell or ovum is correctly fertilized and the process of cleavage starts in the fertilized egg, then it is transplanted into the uterus of the female. So, it can only be transplanted if the fertilization is proper. If fertilization is proper and cleavage starts in the fertilized egg, then it is transplanted in female's body. Then it is transplanted in female's uterus, where it undergoes normal development. Now, test tube baby is a baby obtained by normal development in uterus of the female, but after in vitro fertilization in lab. This point should be noted that test tube baby is obtained by normal development in uterus of the female, but after in vitro fertilization in the lab. The first test tube baby of the world was Louis Joy Brown. Louis Joy Brown was the first test tube baby of the world, who was born on 25th July 1978 in Britain. However, the first test tube baby of India was Harsha. Harsha was the first test tube baby of India, who was born in 1986. Now, this was all about the test tube baby or in vitro fertilization. let us discuss something about the story of Dolly. Story of Dolly. Now, children, you'll ask me, Dolly, who was Dolly? Actually, Dolly was a sheep and it was a clone of a sheep. So, through this story, we are going to learn about the cloning procedure. Now, what happens? Cloning is what? It is the production of an exact copy of a cell or any other living part, or it can be sometimes complete organism also, or complete organism. Now, cloning of an animal was successfully performed for the first time by Ian Wilmot and his colleagues at the Rosen Institute in Edinburgh. So, first time successfully cloning was done by Ian Wilmot and his colleagues at the Rosen Institute in Edinburgh, Scotland. What they done? They have successfully cloned a sheep. They successfully cloned a sheep named Dolly, and Dolly was born on 5th July 1996, and it was the first mammal to be cloned. Now, how Dolly was given birth? Let me explain this to you. So, Dolly was given birth by a Scottish blackface Eve, and it was found to be absolutely identical to the Finn Dorset sheep. It was exactly identical to the Finn Dorset sheep, from which the nucleus was taken. Since the nucleus from the egg of the Scottish blackface Eve was removed, now what they have done was they have removed the nucleus from the egg of Scottish blackface Eve. Dolly did not show any character of the Scottish blackface Eve. Dolly did not show any character of the Scottish blackface Eve. So actually Dolly was a healthy clone of Finn Dorset sheep, and it also produced several offsprings of her own through normal sexual means. But unfortunately, Dolly died on 14th February 2003 due to some lung disease. So now let me explain this cloning process again to you children. And Dolly was the first mammal to be cloned. Actually, there were two types of sheep taken for cloning: Finn Dorset sheep, mother cell, and Scottish blackface Eve mother cell. So now the nucleus was taken from the Finn Dorset sheep, and this denucleated egg cell was allowed to fuse, and then this nucleus became diploid in number. This nucleus ultimately forms cleavage, and due to many cleavages, the nucleus, it was implanted in the surrogate mother. That multinucleus stage was called as blastula in the surrogate mother, and ultimately the clone, that dolly, was born. It unfortunately died due to some lung disease. So this was the story of the dolly and the process of the cloning. Thank you, children.